And now it's time to move to the next speaker, who is Dr. Laven, who is professor and head of the Division of Reproductive Endocrinology and Infertility of the Erasmus University Medical Center. He was initially trained and certified as in obstetrics and gynecology, but then he became a subspecialist in reproductive endocrinology and infertility. For many years, he has had a particular interest in the management of PCOS, covering the full spectrum of the syndrome during woman lifespan, including menstrual cycle, fertility, body weight, and health. And more recently, he has been focused on long-term health risk, as well as the genetic basis of the disease. He has published over 350 papers and several reviews, and has delivered over 300 lectures to international scientific audiences. He is a visiting professor at the University of Shandong in China and at the University of Adelaide in Australia. He is also a member of different international guideline groups for PCOS. So it is a pleasure to have you here, Professor Laven. So if you are ready, you can talk with us about genetic basis of PCOS. Thank you so much.
Thank you, Manuel. So first of all, I would like to share some disclosures with you, and especially the ones marked in red here are of importance and are related to what I'm going to say in the next 30 minutes or so. And thank you, Lisa, for the nice introduction about PCOS in the beginning of your talk. And I learned a lot of new things of the rest of your talk as well. What I would like to do is to take you a little bit on a kind of historic journey through the genetics of PCOS, because we know that PCOS runs in families. And there are quite a few studies which have shown that, and also recent ones. But I would like to commemorate one of my best friends, Rick LeGros, which really was one of the first who published about this. And they looked at testosterone levels in sisters of probants which were diagnosed with PCOS. And they could see that in these sisters, the free testosterone levels showed a kind of bimodal distribution, or trimodal distribution, actually. And that was different in controls, which were sisters from unaffected PCOS probants. And when they focused on testosterone levels per se, they could see that the probant had the highest levels, but the PCOS sister had similar levels, as is shown here in red. And the hyperandrogenemic sister, which had not other signs of PCOS, had also higher levels of testosterone. And that was not the case in the unaffected sister and in controls. And the same was true for DHES, and it was also true for LH, although FSH levels were similar in these women. By far the best study came from a Dutch group which was looking at PCOS incidence and prevalence in dizygotic and monozygotic twins. And they came up with this model in which they looked at additive genetic factors marked as A here, common environment factors marked as C here, and non-shared environmental factors marked as E here. And they could calculate that about 80% of PCOS would be attributable to additive genetic factors. And that some interaction, as Lisa also pointed out to you, is due to environmental interaction with these, probably with these genetic factors. And what they also did in the lower part of this diagram, they looked at whether oligo and amenorrhea, so ovulatory dysfunction, and acne and hirsutism, how they related to these additive genetic effects and environmental effects. More importantly, they showed also that if they looked at, for instance, at PCOS per se, they could see that there was a high concordance of the incidence of PCOS in monozygotic twins, whereas this was not the case in dizygotic twins, which is a very strong indication that it not only runs in families, but they are sharing a big part of their genetics in monozygotic twins. And you would expect that all the features of PCOS, like PCOS itself, but also oligomenorrhea, hirsutism, and acne, which is shown here, that they are much more concordant in monozygotic twins compared to dizygotic twins. And that was the case. Then the next era was an era in which we looked at linkage analysis and also at candidate gene approaches. And you look then at the usual suspects, I would say, and the usual suspects are the ones which are involved in gonadotropic control of the ovary, like FSH, LH, and their receptors, or AMH, genes which are involved in regulating steroidogenesis, like all the CYP17, CYP19, aromatase, for instance, and also at insulin resistance, because as Lisa has pointed out to you, there's a lot of metabolic trouble in women with PCOS. This is a study of our own group in which we looked in 2003 already at different FSH receptor polymorphisms. And we looked at two types, and to cut a long story short, the SER receptor, which is pointed here as SER, is the least sensitive one, and the ASN receptor is the more sensitive one. And one would expect, because anovulation is much more present, and if you give women with PCOS some FSH, that the FSH receptor might be involved in, at least in the phenotype of women with PCOS. And that was actually the case, because the least sensitive receptor was much more prevalent, as you can see here on the left-hand side in red bars, was much more prevalent in PCOS women than in controls. And then we looked at whether these receptors had also impact on the levels of FSH. And indeed, that was the case, because the least sensitive receptor had the highest FSH, endogenous FSH levels, indicating that the body was trying to correct for that least sensitive receptor. And surprisingly, also LH, which is shown here on the right-hand upper diagram, was also higher in women with that SER receptor. And that was related, of course, with testosterone levels as well. We also looked at whether this FSH receptor polymorphism, along with an LH receptor polymorphism, had impact on FSH serum levels, and that was indeed the case, because the number of variant alleles, when it increases, then you could see that the median FSH level was higher the more variant alleles one was carrying. Whether that also was associated with clinical outcomes, we looked into that as well, and we looked at cumulative pregnancy rates. And you can see here on the left-hand side for the FSH receptor, and one would expect that the more sensitive receptor would lead to an earlier response of these women when they were given FSH or clomiphene citrate. And actually that was the case, because what you could see is that the least sensitive receptor had the lowest cumulative pregnancy rates. And when we combine that with some of the genetic variants of the FSH beta gene itself, we could even see that these discrepancies were more pronounced, and that the combination of the more sensitive FSH beta unit combined with the most sensitive FSH receptor had the best pregnancy rates. The problem with all these linkage studies is that they are seldomly replicated in a different unrelated sample to the one in which it was discovered. That was also the case for FSH, because it has only replicated in one small study. The same holds true for the rare variants which were detected in AMH. And although what is shown here on the left-hand side in the orange bars, you can see that these AMH variants were much more common in women with PCOS, or were only restricted to women with PCOS, indicated here as cases. But on the other hand, some of them were also occurring in controls, and some were also occurring in cases as well as in controls. And when one focused on the ones which were only occurring in PCOS women in cases, then you could see on the right-hand side, if they looked at the level of AMH in blood, and that was the reference was used, that it is the gray bar, and that is the homozygous reference with the normal AMH gene, and compare that to the variants. One could see that some of the variants which were in controls, they were similar to the reference, whereas the ones which were much more prevalent in PCOS women had lower AMH levels, actually indicating that AMH could have a lower effect in women with PCOS compared to controls. Insulin receptor variants, there are different insulin receptor variants. There are 3 of those actually which have been studied extensively in different studies, which is shown here. And to cut a long story short there again, most of the time this could not be replicated at a significant level, which is shown in the lower diagram. And that holds true for all the 3 most studies insulin receptor variants in PCOS. And of course, the problem with all these linkage studies is that you expect that there is an effect of one of these genetic variants because you look at the physiological system, and you look at genes which are regulating this physiological system. Then in the early 2000s, genome studies came around the corner. Sorry. What do genome-wide association studies do? They do not have a fixed hypothesis about the effect of some of the genetic variants on the trait you're studying, in this case PCOS. So they are hypothesis-free, and that's an advantage compared to the non-hypothesis-free studies on candidate genes. And this is the first study which was published in 2011 by the group from Zhijiang Chen from Shandong University. We very much collaborated with each other, and I hoped to be the first to publish the first GWAS study on PCOS. But she was a little bit earlier than we were. And actually she published 2 studies in the same year in quite some large sample-sized cohorts of women with PCOS versus controls. And they found at least 3 genome-wide significant hits. And that has to do with numbers, because thereafter the numbers increased, and this is a study which we published back-to-back. On the right-hand side, a study from our group, on the left-hand side, another study from the Dunaive group from the US. And as you can see, the numbers of patients were already a little bit higher than in the Chinese study. And that very much explains why we had more hits in the Manhattan plots here. And that was actually the reason that we joined forces and we came up with the International PCOS Consortium, in which we combined data from different populations like 23andMe, Decode, the Rotterdam dataset, the Chicago dataset from Andrea Dunaive, Oxford datasets, Boston datasets, and datasets from Estonia. And basically, this is the latest GWAS which we published, in which we identified more than 32 genetic variants which are associated with PCOS. So what lies ahead is to see whether we can, let's say, correlate these genetic variants also with PCOS in functional studies. Looking a little bit more into what factors are very much associated with these genetic factors, that's actually AMH and the which is in a way astonishing, because we would expect that things like LH and total testosterone and SHBG, for instance, or fasting glucose would be correlated very much with the hits which we found. And it was not the case. It was AMH actually, and even more pronounced age at natural menopause, indicated as ANM here. So PCOS seems to be very much associated with these two factors, much more than, for instance, BMI, fat-free mass, waist to hip ratio. And last but not least, LH, SHBG, and testosterone are not that very much correlated with most of the factors which we find. So that's a kind of challenge which lies ahead, to see how we could combine all these different results and see how we can build up a kind of pathophysiological system which fits into these genetic variants. Another way to look at that is making genetic risk scores. So what you actually do is you look at the variants which are distributed amongst controls and PCOS women, as is shown on the right hand side here. And then you can make a kind of risk score which indicates then the risk for PCOS. And if you look at, if you distribute them in quartiles, for instance, then if you're in the fourth quartile, if you have 12 to 15 of these genetic variants, then you have an odds ratio which is about 6 times higher than to develop PCOS compared to controls which do not have these much risk factors. And what this study also showed is that it is also related to free testosterone, menstrual numbers, so the numbers of menstrual cycles, ovarian volume, and ovarian follicular number. And here you can see that again, there is a distribution which is shifting to the right in the red bars, indicating that women with PCOS have higher genetic risk scores for this in this rate. If you look then at whether these genetic risk scores are also correlated with free testosterone, for instance, the green line here, then you can see if you run on the x-axis from the left-hand side to the right-hand side, you increase the number of genetic risk score, and you see that that is also associated with higher numbers of free testosterone, with a lower number of menstrual cycles per year, with an ovarian volume and ovarian follicle number which is higher. Another way to look at that is to look at the most significant hits, which is summarized here on this slide, and see how they are interrelated with other factors like testosterone, LH, FSH, insulin, SHBG, and DHEAS. And the more these points are on the outskirts of the polygonal diagrams here, the more this factor is related to the gene which is on top of the polygonal. The interesting thing was that in the International IPCOS Consortium, we combined a lot of data. And for instance, the 23andMe data were data which were very much only for a very small number of patients related to a confirmed PCOS diagnosis. But the majority of women in 23andMe, they just self-reported that they had PCOS. And comparing, for instance, very well phenotyped cohorts like mine in Rotterdam and the Chicago phenotype, Chicago cohort, which is indicated here as NIH and non-NIH Rotterdam, then you could see that most of these genetic variants were similarly distributed amongst these different phenotypes. So they were not at all capable of dissecting out the more well-phenotyped PCOS women from the ones which were self-reported. There was only one factor which was significantly different between my cohort and the Chicago cohort, but all the other ones were not significantly different. And that's strange in a way, because you would at least like to see that they are very much more powerful discriminating between the more well phenotyped women compared to the ones which were less well phenotyped. And that brought us to functional genetic studies in PCOS. And this is one of the first studies in which we looked at DEN-D1A. And that was one of the major hits in the first GWAS analysis. And there are some rare variants in these in DEN-D1A. And they are indicated on the left-hand side by the yellow arrows. So these yellow arrows are genetic variants in that DEN-D1A gene. And when we looked at these DEN-D1A rare variants, and we looked at how they were distributed amongst women with PCOS, with these variants and the ones which did not have these variants, then we could see that LH-FSH ratios were the highest in the women with PCOS which had these DEN-D1A rare variants. And if the rare variant was not there, then LH-FSH ratios were a little bit lower. And that was also the case in unaffected siblings which had rare variants and unaffected siblings which did not have these rare variants. And I have to admit these numbers are very small, but they come from studies of informative trios. And informative trios are very hard to establish in large numbers. But you can see a definite trend here, which is also significant between the ones which had PCOS and had rare variants compared to the ones which did not have these rare variants. Then they looked into whether this DEN-D1A, which had actually two variants, variant 1 and variant 2. And if you look at variant 2, that seems to be the one which messenger RNA-wise was much more found in women with PCOS. They had higher levels of these variant 2. And it was also much more expressed in TICA cells of PCOS women. When you looked at the left-hand side here, there you see a normal follicle with a normal TICA cell line. And if you look at the lower part of that diagram, you can see a follicle of a PCOS woman in which the TICA cell is much more expressing this type 2 variant of DEN-D1A. And there was also that it was much more excreted through the urine in women with PCOS. And if you look at these DEN-D1A variants, you could also see that DHEA levels in the ones which had not these variants had lower DHEA levels and lower testosterone levels compared to the ones which were having these rare variants. So there seems to be a relationship between DEN-D1A, this variant 2, and the phenotype of PCOS in terms of theca cell dysfunction and androgen levels as well. And the same holds true for these AMH variants, which I showed you already in a previous slide. And there they looked indeed at whether these AMH variants had impact on CYP17 and on CYP19. And actually they had impact on CYP17 messenger RNA expression, because all these orange bars, except for one, show that they have definitely lower levels of CYP17. And that takes you to the right-hand side of this slide, in which you see on the left panel a normal ovary and on the right panel a PCOS ovary, in which these AMH variants apparently do not, let's say, inhibit CYP17 that much, and they are associated with increased CYP17 levels, and therefore actually they at the end of the day lead through DHEA and androstenedione to a higher level of testosterone in these TICA cells. So there is a functional relationship between these AMH rare variants and the phenotype in terms of testosterone production in TICA cells. Another interesting finding was, I showed you already, that PCOS variants were very much associated with age at natural menopause. And this was a study which we did in 2018. And in 2016, we also reported on that already. And there you could see the effect on AMH levels and the effect on menopause. And in the green area, you see the genetic variants which are clustering in that area, which have large effects on age of menopause. And it seems that PCOS is associated with a kind of selective enrichment of menopause postponing genes. And it makes sense because we know already from large epidemiological studies that indeed PCOS women generally enter menopause somewhere between 53 and 60 years of age, and they have an average age at menopause of 56, which is 5 years higher than what you would expect in the normal Caucasian Western European population, because their menopause starts around age 51. In our recent study, and that's unpublished data, and please do not take any pictures of this because otherwise I get into trouble with my co-authors. There are a few other slides, and I will mention them, where you are not allowed to take any pictures of that. This is a genome protein analysis, and again, you have a kind of Manhattan plot at which some of these proteins pop up, and they are significantly associated with PCOS. And that's even a more complicated approach, because then you have to look at the genomic region which is associated with these different proteins. Then you look at the proteins which are upstream driven by these factors, which is shown on the right-hand side here. And then they might impact through either BMI or directly on PCOS. And there are also some downstream proteins which might have influence in PCOS in that way that they might exaggerate the phenotype. But there is a lot of work still to do in this respect, because this is only the beginning of a very complicated mathematical endeavor to see which factors are very much associated. A way to get around that is phenome-wide association studies, and that's called PheWAS. And what you do actually in PheWAS is you look at the phenotypical features, and you try to combine those to make it a little bit simple, to combine those with genetic risk scores and develop an algorithm which is then predictive for PCOS, and then do in a large population, and most of the time they do that on electronic health records, in a large population of normal women and women with PCOS, which are, let's say, in that population, and then see whether this genetic combined PheWAS score tells you something about the phenotypes which are associated with the trait under study, in this case PCOS. And another way to do that is Mendelian randomization. And in Mendelian randomization, you try to figure out what is causing PCOS, what does PCOS cause, and what is not associated with PCOS or is not causally related with PCOS. So this is the PheWAS study, which was published in 2020. And to take you to the left-hand side, it was done in 49,000 females. And then they looked at how what PCOS features were associated phenotypically with all kinds of different diseases. And what you see there is that polycystic ovaries were associated significantly, but also diabetes, hyperlipidemia, type 2 diabetes, obesity, morbid obesity, disorders in lipid metabolism, sleep apnea, hypertension, diseases of the esophagus, phlebitis and thrombophlebitis of lower extremities, and essential hypertension, which makes sense because actually we knew that already, because we have phenotypes which are more metabolically disturbed, and we have phenotypes which are more reproductively disturbed. They did the same thing in 41,000, nearly 42,000 males, and there was no association of these factors found except for type 2 diabetes and morbid obesity. And on the right-hand side, you can see the phenotype groups which are associated. And looking at the pie diagram here, you can see that endocrine and metabolic issues are very much associated, digestive things, and circulatory system is also very much involved in PCOS phenotype-wise. Mendelian randomization, I told you already. Then you try to figure out what is caused by what is causing PCOS and what does PCOS cause. And there again, we found similar things in terms of SHBG, testosterone, free androgen, age at menopause, childlessness, but also the more metabolic traits, which is in the upper part of this diagram, they are also very much associated with PCOS and have impact on PCOS. They might be related in some way or the other and cause PCOS. We also looked at heterogeneity between sexes, whether that was different in males and females. And there you can see that it's definitely driven in the female phenotype, but there's also some of these are partially driven by the male phenotype. As Lisa already alluded to you, it's not very well known whether these things are associated. And this is a very nice study from Mark Udasi's group, and they looked at all the green metabolites in the left upper corner. There is evidence that they have a causal effect on PCOS. So they might cause PCOS. And the ones on the orange upper box there are showing evidence that PCOS may cause these outcomes. And there's a lot of things which are not caused by PCOS, like high BMI, like type 2 diabetes, like cardiovascular disease, like stroke. They are associated with PCOS, but not caused by PCOS. And there are also a lot of factors which we generally would like to, let's say, incorporate in a causal way to PCOS, which are not causally related, according to these Mendelian randomization studies. The latest approach which we took is cluster analysis. So we did an unsupervised cluster analysis in which we looked at insulin levels, BMI, glucose, DHEAS, testosterone, FSH, LH, and SHBG. And looking at how they were clustering into a metabolic phenotype and a reproductive phenotype, you could see that the metabolic phenotype was very much driven by differences in insulin, BMI, and glucose, which is shown in the lower left-hand corner here. And the reproductive phenotype was much more driven by the FSH, LH, and SHBG levels. Surprisingly, testosterone and DHEAS did not differ between these two phenotypes. And what this group for the first time did, they did a GWAS in these different reproductive and metabolic phenotypes. And what they could see is that they had different genetic hits in the reproductive phenotype compared to the metabolic phenotype compared to the indeterminate phenotype. Which is the first time that we saw that a phenotype is very much associated with a different genetic background. And that's actually what you would like to have, because you would like to have a disease which is not only phenotypically different, but also genetically different. Then we took this a little bit further and we looked in our Dutch cohort whether that was also the case. And actually, if we looked at NIH criteria, comparing with the American approach in the previous slide, then you could see again that we had a similar distribution of the reproductive and the metabolic phenotype. And we also saw that there were differences between the metabolic phenotype in a genetic sense and the reproductive phenotype. And that was also the case when we used our own Rotterdam cohort, which included much more different phenotypes, A, B, C, and D, compared to the American cohort where only NIH criteria were used to diagnose PCOS. And there we could see the same thing. We had a little bit more hits, but that is again determined very much by numbers. We also looked at, when we looked at metabolic phenotypes and reproductive phenotypes on the left-hand side here, and how they sequestered into the phenotypes according to the Rotterdam criteria. And then you could see that some of them shifted from the reproductive and the metabolic phenotype to the background phenotype. But the majority of the background phenotype women, they shifted in the Rotterdam criteria to the metabolic and to the reproductive phenotype. And then we did something different. This is again, and it's another study because we were not the only ones which were looking at cluster analysis. And what they actually showed is that they found similar clusters as ours, metabolic, reproductive, and they called the indeterminate the balanced one. And what they also looked at is on the right-hand side here, in the upper corner, they looked at phenotype A and how that sequestered into these three different metabolic phenotypes, indicated by the red lines. And for phenotype B, it's indicated by the blue lines in the right upper corner there. And in the lower right corner, it is phenotype C, which sequesters in a different way. And that is also the case for phenotype D. So apparently it depends very much not only on the phenotype, but also how you sequester into these different clusters. And then we did something, we developed genetic risk scores for these different clusters. And this is the metabolic genetic risk score. And the genetic risk score was distributed here in 5 quintiles. And if you look at the 4th and the 5th quintile, they virtually are 100% metabolic phenotype. So they are actually capable of discriminating the reproductive and background phenotype from the metabolic phenotype. And to cut a long story short, we did it for the reproductive phenotype as well. And there again, the reproductive phenotype perfectly sequesters apart from the metabolic and the background cluster. And there again, you can see that it also holds true for the background phenotype. So this is the first data which is showing that the metabolic and the reproductive phenotype of PCOS are genetically also distinct and different from each other. And this is the first time that you have some information available which you could use, as Lisa was alluding to you as well, as a kind of driver for treatment, because they definitely make a difference treatment-wise as well, as I think, and also in terms of long-term health aspects. And we are very much looking into that at the moment, whether they are correlated with these outcomes as well. So what are my take-home messages? PCOS runs in families and the heritability is estimated to be around 75%. Cysts are either affected or unaffected, as well as brothers, fathers, and mothers share PCOS characteristics such as high AMH and high steroid serum levels. Early SNP studies suffer from a lot of flaws in terms of numbers, population stratification issues. Results from were often inconsistent and rarely replicated in independent samples. But it seems that FSH receptor, FSH beta, insulin receptor, and AMH variants have been replicated and are involved in some way in the pathophysiology of PCOS. G was revealed up to now some 30 loci associated with PCOS and some 30 proteins related to these genetic markers. Some of the identified loci from GWAS are strongly associated with metabolic, whereas others are more impacting on reproduction. GWAS also has indicated that PCOS is associated with delayed ovarian aging and later menopause, and Mendelian randomization indicated numerous factors that are causally related to PCOS. And last but not least, cluster analysis identified clusters with different genetic backgrounds, and they might impact in a different way on reproductive and metabolic phenotypes and provide ways for individualized treatment of women with PCOS in the near future. Having said that, this is the team which makes all the work possible, and I am the professor who is telling the story, but they really need to be acknowledged. Thank you for your attention.
So thank you very much, Professor Laven, for this very interesting presentation also. Now we move to the final part of this session. So we are running a little bit longer than expected, but we still have time for some questions. Indeed, well, remember you can send the question through the Q&A button, but we still have some of them. They were already sent for you, Professor Stenner. At the end of your talk. The first of them is from Christian Omoage. He is asking, he's saying metformin is safe in pregnancy. So do you think that metformin use preconception and throughout pregnancy will reduce the risk of transmitting PCOS to the offspring?
Mm. First of all, as I'm not a clinician, I don't want really to talk about recommendation, but one thing is that I'm not 100% sure that we can today say that metformin is safe during pregnancy. There are, especially there, Esther Banki have done several randomized controlled trials and also followed the offspring. There she actually see that metformin do not make the women less likely to develop gestational diabetes or miscarriage, or it does not really make everything better during pregnancy. And she also see in the offspring that when they grow a bit older, they develop more metabolic symptoms. They are being more obese. She have also measured different things like head circumference is larger and so on in offspring with metformin. And I also want to draw the attention to diabetes field, because there are now researchers working with mice model with diet-induced obesity and so on. There it seems that maybe during pregnancy per se we should still not be say that it is safe, but from our study I would say that pre-treatment, I think that is something we should maybe do, and that is also the study by Laure Tappanen or Laure Poppanen and Juha Tappanen. What they show is when they pre-treat. So I think that is what we should do right now, and then we really need to understand whether it's good or bad to use it during pregnancy. Do you want to chip in, Joop?
No, nothing to add to that, Lisa. And indeed, we should be a little bit careful, because it seems that metformin use during pregnancy is associated with some differences in offspring, to put it very carefully. But yeah, and it's used a lot across the world, also during pregnancy, actually.
Yeah, for type 2 diabetes, everyone is on metformin, yes. So
yeah, there is another question related with metformin, but I don't know if this is the good place or good forum to answer. Is I don't know if you want to say something, because it's
yeah, there I see that is whether we can start with metformin when HOMA is between 2 and 2.5. And I think in our study, all women, we didn't set a cutoff on the HOMA. All women were had a BMI above 25. So there were women with lower HOMA. And I think there I have no really how to say bad feelings that you actually can treat women that has a lower HOMA, because metformin, we know today that metformin is not just a drug to improve glucose metabolism. We know that it is used as an anti-cancer drug. It has many effects that we don't really know what actually the mechanism is. And I think our single nuclear RNA sequencing study really highlights that it has effects on certain cell types that we still don't understand, that we need to look into much more.
There seems to be also a very strong anti-aging effect there.
Yes.
I would like to add something to that. The more we look into this metabolic phenotype, also in a genetic sense, it seems that, and that is very much driven by insulin and by glucose levels and by BMI, that might be a different kind of type 2 diabetes, which is running in women with PCOS compared to what is seen in the general population. I am saying that because we did a large randomized controlled double-blind study in which we tried to reduce the number of gestational diabetes in women with PCOS which were pregnant by using myoinositol. So half of the population, 450 women, got myoinositol and the other half got a placebo. There was no difference at all in the incidence of gestational diabetes, which is around 11% in that population, and it was exactly the same in the population which was using myoinositol. So also myoinositol, along the same line as metformin, is apparently not able to reduce the incidence of gestational diabetes. Whereas out in the wild, in people with diabetes, it is. So they probably suffer from a different form of diabetes, in this case gestational diabetes, which is not responding to metformin or myoinositol in a similar way as it does in diabetes per se.
Yeah, okay. What about the calorie restriction, the study that you saw? Because probably can be some shared mechanisms, right?
Definitely. I think it is also quite important to highlight here. I cannot really recall, but it was quite a long time ago. Rob Norman in Australia, they did a caloric restriction study prior IVF, but they did another kind of caloric restriction. It was this very low calorie diet that is really put them into a catabolic state, and if I remember correctly, they had to stop that trial because that had detrimental effect on the oocyte quality and so on. So, but here they did a caloric restriction that they still kept the amount that they eat, but they lowered the calories. And again, we know that it is not bad to actually lose weight and keep your diet. That is the first line treatment for women with PCOS. And if you are overweight and obese, I definitely think it is the way to go, maybe together then with metformin or other glucose sensitizing like GLP-1 or receptor
Your incretins
Yeah.
might be very helpful there again, because in general, we did a large randomized trial on lifestyle intervention, and that was repeated by many others, and we did a recent meta-analysis. And lifestyle intervention is not that easy in women with PCOS, and the effect size is really very, very, very small, actually. And so we should not bother our patients with, let's say, extensive lifestyle intervention programs, because they are generally not very effective. I believe much more in bariatric approaches or as Lisa was showing, incretins, GLP-1 and GIP and combinations of that. And actually, we really should focus on those, whether that might help our patients much more than.
Yeah. Okay. So yeah,
And I can also add that lifestyle management in our recent study, it was a very mild one. And despite that, we could see that also in the same subpopulations in the cell types, we could see a positive effect in the lifestyle group only, which I think is also very promising.
Yeah, and maybe Lisa, we share the same idea about that, because I got the impression by switching from a kind of anabolic state into a slightly catabolic state without losing that much of kilos, did already in our initial pilot, which we did after Rob Norman did that study, showed already that insulin resistance completely disappeared within 3 months. And then these women did not lose, let's say, like 5 or 10 kilos. They lost just 1 or 2 kilos.
I think it is a change also. you know, if you have like a steady state, you need a change. It does not need to be something very dramatic. And I think
Yeah.
it is a very important message also to these women, because they suffer and they really try hard and think that everything is and then they blame themselves that they cannot make it and so on. So
Exactly.
it's an important message for clinicians dealing with these women.
Yeah, it seems that cells and bodies get used to the changes. No, then we have to challenge them continuously to make them move.
Yes.
Right?
Mm.
Exactly like that.
Mm.
Well,
And we should follow our women. I think that is something we are bad in. They should be followed.
yeah.
Mm.
Yeah, well, it is clear we are learning a lot about PCOS, but I think there is still a lot of to learn about the epigenetic, genetics, also transcriptomic. I saw splicing in your presentation, Professor Laven, also proteomics about the associated with the phenotypes, also with the transmission from a mother to the litter. So, but well, I think with your presentations clearly illustrated that we are in the way to trying to understand all these changes. So I think we can close the session. Thanking again both of you for accepting our invitation to be here and for sharing all the information or your knowledge. I think it was a really, really interesting session where we learn a lot. So thank you so much for being here. We will see you next time in the next Spotlight of Science that will be on September and the last one of the year in November. Thank again to all of you for being here, to Recordati Rare Diseases for supporting this educational calendar. I hope see you soon. Really nice summer for all of you. Thank you so much for Professor Stene Victorin and Professor Laven for this great presentation. Thank you so much. Bye-bye.
Thank you.
Bye-bye.
Thank you.